much for this wonderful introduction. It's a great pleasure to be here. Thank you to the organizers for making that possible. I hope to be able to contribute a bit by sharing with you our efforts and our results on bridging the gap between cortical structure and cortical function. My talk will have 3 parts: an introduction, and then I will summarize quantitative anatomical studies and conclusions thereof on cortical function. This is work over many years together with Valentino Breitenberg, former work, and this will be the main part of my talk. I apologize to those who know this already, but I think it is relevant to the topic of this meeting. Then in the last part I will very briefly talk a bit about the connectivity of the cortex in its horizontal plane, which is interesting for people who are think about the spread of excitation over the cortex. Now, when we think about cortical functions, we come up with terms like learning, thinking, associating, perception in the different modalities, recognition, control of voluntary movements, language, categorization, calculating, composing music, and other wonderful higher cognitive functions. On the other hand, when you sit over the microscope, you see something like this, and you wonder how you ever will be able to get from there to something, to these functions which I just mentioned before, and there seems to be no cue, no way to get from one to the other, although it is clear that somehow these functions must be hidden in this structure. Let me show you a few more examples of classical anatomical pictures for those of you who are not looking into the microscope every day. Now this here is a classical Nissl stain which stains cell bodies. This is a vertical section through the cortex, and you get an impression of the density of neurons there and of the layering. Another classical stain is the Golgi stain. This is a slider. We go now up in magnification a bit in the next slides also. This stain selects a few neurons by chance and stains them with all their cell processes. So you see here about 10 neurons with their dendritic trees, and you see some axons in the background. And if you superimpose in your mind this picture with the previous one, you can imagine what an enormous jumble of cell processes you have in the brain, in the cortex. And this stain gives a more direct impression of this. This is a Bilchowski stain, which stains axons. This is a thin section of about 30 micrometer, and it is already nearly black because of the high density of axons, except where there are cell bodies in the background. So some of the fibers you can resolve, others are just merged here. And questions come up, for example, does this fiber know where it has to go, or is this also partly a matter of chance? Now, if you go still higher in magnification, go to the electron microscope, you see something like this, and you see really the enormous compactness of the structures. So I think this, since we are comparing brains and computers, this is something which is probably quite different from electronic devices, to have this enormous intertwining of processes as it is here. Now, this is part of a nucleus, just to get an impression of the magnification. All what you see here are cross sections and length sections of dendrites and axons, and a few glia cell processes. In particular, you see here also the synapses. You recognize them by the black postsynaptic thickening. So this is a postsynaptic element. And on the presynaptic side, you see a lot of transmitter vesicles. So here is a synapse, and here, and where's the red dot here? Or doesn't work very well. Okay. Anyhow, you see in the microscope, you see everything you need, everything which you need to see for. You see all the conducting elements, and you see the transmitting elements from one neuron to the next. But still, you do not understand what the link is to these functions mentioned before.
There's another pointer right there.
Yeah, thank you. Maybe, yeah. Now, one thing is striking, namely, on the one hand, you have this variety of functions, which I mentioned before. On the other hand, you have a relative homogeneity of structure. All of these pictures, except the first one, could have been taken from everywhere in the cortex. The first one, the Nissl picture, shows some variation, local variations, but they are very subtle, and in most cases very subtle, and you need some exercise to recognize them at all. So this suggests, as has already mentioned this morning in Dharmendra Moda's talk, that maybe this variety of functions is based on one or a few basic functions. And if it is possible to understand the basic structure of the cortex, there may be a link, at least to these basic functions, and this may help to link the two things. Now the problem is, what means to understand the structure? This means to understand the network structure. This is something, how the neurons are connected. This is something you do not see directly from these pictures I just showed you. So our approach was quantitative anatomy. So we measured the various things you can see in the microscope. This is a project which was started quite a long time ago by Valentino Breitenberg, and in which I joined him in the 70s together with some other co-workers, Günther Palm, who is now in Ulm, and Bernhard Hellwig and Horst Greilich, who were PhD students at the time. And so what we did was to measure various things in the what you can see in the microscope. You don't go in the details here. It's just to show you the approach. From these measurements, you can deduce some other quantities, and this indeed gives you some interesting insights into the network structure of the cortex and the possible functions behind it. Now, I want to go through the most important points of this table in a few stepwise. One of them is evident. One of these points is evident from this picture. There is, at least in larger brains, you have an enormous amount of white matter, cortical white matter, which is stained black here. And only this bundle here, the internal capsule, carries fibers which connect the cortex with other parts of the brain. And you see this is very small compared to the whole amount of white matter. So this must be fibers which connect, mainly fibers which connect the cortex in itself. And this can be quantified by estimating the number of input neurons to the cortex. For from other parts of the brain, and they are of the order of 10 to the 8th, while the neurons in the cortex, as is known from work by Haug, for example, are of the order of 10 to the 10th. So most of the synapses in the cortex are synapses between cortical neurons. So this is the first main point here. Another point is when you look at the cell types and don't look at them in the detailed and wonderful way as it was done by Ed Callaway this morning, but in a more rough way. You see that most of the neurons are of one main type, namely pyramidal cells, about 85%. This is data which came first from the lab of Peters. Now, how are pyramidal cells defined here? Pyramidal cells are characterized by having many dendritic spines on their dendrites, which are the postsynaptic structures of pyramidal cells. So on each spine sits a synapse. Pyramidal cells are also characterized by having both local and distant connections. So the axon makes local axon collaterals in the environment of the neuron, and the main axon then goes down to the white matter and makes a terminal ramification somewhere else. And to put all the neurons together into one main class, in spite of the lot of variations among them in shape, makes much sense because all of them are excitatory. And so the next important conclusion is that the vast majority of neurons is of one main type, namely the pyramidal cells, among which we include here the spiny stellate cells, and all of these are excitatory. After that, it is not astonishing to find in electron microscopical investigations that most of the synapses in the cortex are also excitatory. About 90% of the synapses are excitatory synapses. Classifying the synapses further, you find that the majority is located on dendritic spines, which I just showed you before. So about 75%. Now, so these are all located on pyramidal cells, and the excitatory synapses are made by pyramidal cells. So this leads to the inevitable conclusion that most of the synapses in the cortex are synapses between pyramidal cells. Now, why are so many synapses on spines? What is the role of spines? Synapses can as well be located directly on a dendrite. So there have been various theories about it, but there is quite some evidence that spines have to do something with learning, that they are particularly suited devices for strengthening or weakening for the synapses on them by changing their shape. This is something which has been around since the 60s or so, the idea that the shape of the spine may have an influence on the strength of a synapse. And if this is true, and there is more and more evidence for that, one may conclude that most of the synapses in the cortex are learning elements, and maybe this is one of the main, just simply one of the main features of the cortex to be a huge learning device. Now let us come to a few concrete numbers, which I think have been here already this morning. Number of neurons in a cubic millimeter is nearly 10 to the 5th in the mouse, and the number of synapses is nearly 10 to the 9th. And this leads to an average number of synapses of about 8,000 per neuron. Now, an important question for the network structure is, are these always different neurons, or are there connections between many connections between few neurons? The answer is most of these synapses are synapses between different neurons. I don't want to go into the details about that, but let me convince you by showing you this picture here. This shows a pyramidal cell in a cubic millimeter of cortex, and these dots here indicate the density of neurons at that magnification. And it is immediately clear that this local axonal tree, drawn in blue, penetrates thousands of different dendritic trees. Now, so this already suggests that one neuron projects to many other neurons, but you need also to know how are the synapses distributed along the axon. Are they distributed throughout, so that it is likely that they make synapses with all or many of the dendritic trees that they cross? The answer is yes. The axonal boutons are distributed all along these axonal ramifications. From such numbers and some others, one can make estimates on the probability of connections between two pyramidal cells. And this shows one of the approaches we used. So you need only to look at one of them. They show all the same thing, but in different layers. Let's look at this one here, in the lower right here. What you see here is the connection probability on the y-axis, and on the x-axis you see the distance between horizontal distance between two neurons, and you see that when they are close together they have a relatively high probability of being connected, while and this probability decreases with distance in the way shown here. Now, but what is interesting for knowing more about the divergence and convergence, the question, how many multiple contacts does a neuron make? So if you pick out two pyramidal cells and ask what is the probability that they make 1 or 2 or 3 or 10 synapses with each other. If we again look at just one of these pictures here, for example this one, you see that in all cases it is the highest relative probability is that they make only one synapse. So this is the probability for one synapse, for 2, 3. Okay, so the uppermost curve is always for the probability for one synapse, then for 2. Okay. Anyhow, you see that it is the probability that they have multiple contacts, more than 1, 2 or 3, is quite low, in particular at larger distances. So from this, one can draw the following conclusions, namely that an individual pyramidal cell has thousands of synaptic targets, so that there is a high degree of divergence of signals. Also that most pyramidal cells are weakly connected to each other via only one or a few synapses. Oh, thank you.
It's the red one.
Here? Ah, yeah, great. Yeah. And also that each neuron is connected with every other cortical neuron via one or a few synaptic steps. Now, this last point here sounds as plain contradiction to what Ed Callaway said this morning, but I think it is, and I was wondering if I should take it out, but but I didn't because we are here in an audience of computer scientists, and I think the point here is to show what is similar in computers and what is different in computers, between brains and computers, and I think it is clear that even though there seem to be some biases away from a simple statistical scheme between some populations of neurons, the situation is very different from any electronic device where you have where each junction is completely predetermined, as far as I understand, while in the cortex, where each element is connected to thousands of others and again to thousands of others and so on, it is certainly not predetermined in any way which comes close to what you find in an electronic device. Okay, now what is such a network suited for? Now I come back to this frightening table and but we look only at these points here. What I just told you is this is what I already told you. These are the conclusions on the features of this network, characteristic features of the cortical network. And if you ask what can this network do, it seems an important point seems to be that the cortex can be interpreted as a mixing machine, each neuron getting thousands of different inputs. And also memory seems to be an important point because you have many modifiable synapses. We say memory rather than computation, meaning by this that the dominating thing seems to be an excitatory network of pyramidal cells which are connected among themselves via these typical dendritic spines. And these 2 points together qualify the cortex as an associative memory, as with the formation of cell assemblies in a way as postulated a long time ago by Hebb. Now, this Hebb's theory, I think by these findings that the structure of the cortex fits so well to this theory, I think this gives great support to this theory. I don't know, I think I don't need to go into detail about it. I think most of you know this theory, namely that what Hebb postulated was that learning is made by strengthening of synapses between neurons which are often active together. Neurons which are often active together signify some correlation in the outside world. So it can be for the properties of an object, for example. And if this learning process has happened often enough so that these neurons have formed a network among themselves which is stronger than with other neurons, it is enough to activate part of it to ignite the whole set. So if this represents a rose, it may be enough to mention the name rose in order to activate the whole set. Part of the theory is also that such cell assemblies may overlap if they represent objects with overlapping features, such as, let's say, the red one represents a rose and the other one a tulip. There are certainly common features in tulips and roses, so these will be represented by overlapping neurons. So there is the possibility that the activity spreads from one or jumps from one cell assembly to the next, and so on, in a process of thinking. Now, Hebb at the time could not know these details about the structure of the cortex, but if one would think about the requirements which you need for cell assemblies of this kind, you would say that you need many neurons of the same kind which do nothing but sit there and wait who else is active together with me. In order to know that, they need to be connected with each other into a network. They need to be connected via excitatory connections. The connections must be modifiable. They should not only be local in order to connect things of different between different modalities, and each element should be connected to many others in order to detect as much common activity as possible. Now, this is exactly what you find in the cortex, as I just pointed out. A large number of neurons of the same type connected with each other, connected via excitatory synapses, via modifiable synapses, making both local and distant connections, and each pyramidal cell is connected to thousands of others. Now you may say, well, that's a very common statement, and maybe this is true for the whole brain, and this is nothing special, and so on. However, this is not true if you look at the other main parts of the brain, and I think with these 4 parts you have about 90% of the whole brain. The cerebral cortex is the only one which has an excitatory, which forms an excitatory network. If you look at the basal ganglia, I'm now only talking about the main type of neuron in each of these parts. In the basal ganglia, neurons form a network, as they do in the cerebral cortex, but they form an inhibitory network. In the thalamus, the main type of neuron, the relay neurons, are excitatory, as are the neurons in the cortex. However, as one knows from work by Jones, for example, or the interpretations of Robert Miller on it, these neurons do not network with each other. They project to inhibitory neurons, but they do not form a network among themselves. The same is true in the cerebellar cortex, where there are an enormous number of excitatory neurons, but they avoid absolutely to make any synapses among themselves. They only project to Purkinje cells and other inhibitory neurons. So this is really something special. And so if we ask for the very basic function of the cortex. We can say the cortex is what the cortex can do, or probably at least can do better than other parts of the brain, is to deal with correlations, to detect correlations in the outside world, to incorporate them, and to handle them. And these are terms which we usually describe by learning, associating, and thinking. So I think this is really a link between structure and function of the cortex. Now, I cannot talk about these things without mentioning at least this theory, which has already been mentioned today also, namely the thin-file chain theory by Moshe Arboles, which fits also very well to the structure of the cortex. In this theory deals with the question, how can sequences of activity be organized? I don't want to go into that now. I just want to say that this theory is certainly not in contradiction to the cell assembly theory by Hebb. One can say that cell assemblies are the result of coincident correlations, while thin-file chains are the result of time-shifted correlations. Now, for both of these theories, it is interesting to know more about the horizontal connectivity of the cortex, and here I come to my last part. The connectivity of the cortex in the horizontal plane. And the things I told you until now were gained from studies on the gray matter, and now we switch to the white matter. And I will briefly tell you some results from two studies, one on the human cortical white matter, again together with Valentin Breitenberg, and the other one from a tracer study in the mouse cortex, together with two students, Daniel Liewald and Dennis Chaimov. Now, the horizontal connectivity over the cortical plane goes mainly via the white matter. And the question we asked was, what is the number of fibers in the long cortical bundles? Fibers which go over long distances tend to form bundles in the white matter, and which run in the very depth of the white matter. Here in this atlas from Nieuwenhuis, you see the main bundles. The largest one is here this superior longitudinal fascicle, which carries fibers from the frontal lobe to the parietal, occipital, and even parts of the temporal lobe. Here is the uncinate fascicle, which carries fibers from the temporal lobe to the frontal lobe, and there are some others. What we did was to dissect the white matter at places where these bundles are most compact, measure the diameter of these bundles and divide them, the cross-sectional area, and divide it by the cross-sectional area of the average fiber. And by this you get an estimate of the number of fibers in these bundles. This is the result. Again, don't go into detail. This here shows the fascicles we looked at. This is the superior longitudinal fascicle, which I just showed you. These are the cross-sectional areas, and this is the number of fibers estimated in these bundles. And just look at the orders of magnitude. They are of 10 to the 6th, 10 to the 7th, 10 to the 7th, and so on. 10 to the 6th and 10 to the 7th, and this adds up to approximately 10 to the 8th fibers. So these long bundles connecting between the lobes are carrying about 10 to the 8th fibers. This is an interesting number because it is similar to the number of fibers in the corpus callosum, which has been estimated by these people as a few times 10 to the 8th. So we can say that the number of fibers which connect one hemisphere in itself over long distances is similar to the number of fibers it sends to the other hemisphere. We were astonished about this because 10 to the 8th is a low number compared to the 10 to the 10th neurons in the cortex, most of which send a fiber to the white matter. So in a somewhat different approach, we made an estimate on the length distribution of fibers which connect the cortex in itself over the horizontal plane. And we made 3 groups. So we looked not only at the white matter but also at the gray matter. So the compartment A is the fibers which run horizontally within the gray matter. So it's the axon collaterals of pyramidal cells. And they can reach lengths of a few millimeters, as one knows from work by Jones and De Filippi. So approximately up to 3 millimeters, although most of them are much shorter. Now, in the white matter, we made 2 compartments. One is the compartment B is the so-called U fiber system. This is fibers which follow the gyri and sulci. Here, we assumed that the length of the fibers is as much as the depth of a gyrus and perhaps backwards again, so up to about 3 centimeters, while C contains all the rest, the fibers which run between not directly neighboring gyri but more distant ones, and all the other fibers, the long fibers which I just mentioned, and corpus callosum and so on. And you see, at least in case of this rough division, you get an inverse relationship between number of fibers and length of fibers, which means that neighboring regions have a considerably higher probability of being connected than distant regions. Now, the theoreticians ask us questions we cannot answer on the human brain. So for the very last part, let me switch back to the mouse brain and answer 2 questions, namely, what is the global pattern of connections over the cortical surface, and what is the influence of a small column on the rest of the cortex? This is work done with a tracer study, an anterograde tracer, which looks like this. This is a cross-section through a mouse brain, and the tracer is taken up by the neurons and transported to the terminal fields of the neurons. So here we are close to the injection site, and you see how the fibers radiate from the injection site. This is about the size of the injection here. And you see also distant projections, like here in the other hemisphere or here in the same hemisphere. If you make from serial sections of that kind two-dimensional surface maps of the cortex, you get something like this. These are examples from 3 mice, right and left hemisphere, right, left hemisphere. This is the corpus callosum, so the 2 hemispheres would be knitted together here. And you look now on the surface of the unfolded cortex. The cortex of the mouse is anyhow not folded, but on the flattened cortex. Now the white spot shows the injection site and the region of uptake. So the neurons which have taken up the tracer sit in this white spot. And from there you see how the projections are distributed in the same or in the other hemisphere. Now, all these cases look very different from each other, which is not astonishing because the injections are injections into different cortical areas. But they have a few things in common, namely that in each case you have an enormous divergence, a high degree of divergence by more than a factor of 100. The injection size is about, or the spot of the region of uptake has about 300 micrometer diameter, so this is less than 0.1 square millimeter, and so the divergence is very large. And another interesting point is that at least 2/3 of the total terminal field in one hemisphere is in the neighborhood of the injection site, so is only a small part is contained in the distant spots. Now, and this is something the mouse has in common with the human brain, as we just saw. Both, also in the human brain, the majority of the projections does not go very far. And the same seems to be true in the mouse. Now, there are 2 more examples. What the theoreticians like to know, or the simulators, or people who think about the spread of excitation, they want to know the neurons which are sitting here, which are several thousand, do they all project to all these places, or how large are the subsets of neurons which project to one such spot? Because this gives an impression of the strength of connection from one place to the other of projection. Also, what is interesting to know, what is the density of ramification of fibers in these spots? The answer to the first question is, namely, that columns containing several thousand neurons, pyramidal cells, project to individual distant spots via 10 or up to several hundred neurons. So it is always a subset of neurons which, for example, here in this small spot, only about 14 neurons projected from here to there, while here it was nearly 900 neurons. So that's the orders of magnitude. Concerning the density of ramifications in these fields, here is an example for a strong projection with a high density. This is an example for a projection of middle density, and this is an example of a weak projection where fibers go only to one layer and with a relatively low density. And if one estimates the densities, one comes up with such numbers. So it is meters per cubic millimeter. in case of a low density about 3 meters, a few meters, and here about 25 meters. So that sounds a lot. However, this is very little compared to the total density of fiber length in the neuropil. In a cubic millimeter of cortex, you have several kilometers of axons, and even in the dense spots, you have, in case of the dense spots, you have hardly 1% of the fibers are coming from an individual from an individual injection site. The last question is then, what is the fiber density in this black region? What I just showed you is densities in distant spots, but locally, what is the density here? The density locally is always considerably higher, in the range of 100 meter per cubic millimeter, but even there, this is only a few percent of the total fiber felt in such a region. So these are the main conclusions on horizontal connectivity from these two studies. Namely, on the whole, they are in support of the view as the cortex as a mixing machine. You have a great spatial divergence of projections, and at any given place, fibers come from many different sources, so you have also a high degree of convergence at any place. And from both studies, one can see that the majority of projections connect close regions. And this is something we heard also already this morning, I think by you, that this is something which seems to be common to various kinds of networks, communication networks, also in society. The famous small world effect, that you have in such networks, you have a lot of neighborhood connections and a relatively low number of long-range connections, and this seems to be a good compromise between, on the one hand, getting over a few steps from each neuron to every other, and saving a lot of cable, which is certainly important in the cortex. So here, this is obviously also a principle which is realized in the cortex. Now, at the end of our talk, I want to mention some theoreticians with whom we've discussed this, and Günther Palm again, who is in Ulm, and then the group of Ad Arzen, particularly Stefan Rotter and Nicole Voges, and also Fritz Sommer from Berkeley. Thanks to them for suggestions and discussions about these horizontal connections. Thank you very much.
Time for questions? Introduce yourself.
My name is Paul Bush. This morning, Ed Callaway was talking about a probability of connection between two cells that were right next to each other around, I think he said 20 to 30%, whereas your figures, it looked more like 60 to 80%. Would you comment on that?
Yeah, I think you said much less, wasn't it?
Yeah.
You said neighboring neurons are. What was your number?
In the experiments that we did, two cells right next to each other were connected 20% of the time. But I think the data he's talking about is where you plotted the probability that the dendrites and axons overlapped, which was 80%. But you also had the number of 10%, which is closer to our number, which is based on, I don't know how you made that transition between the two.
Yeah. I should say that these curves showed the probability of connection. I mean, it was made by shifting, computer-wise, shifting neurons against each other. So just looking how often, if you have two neurons, how often do they fall into the same pixel? How often can they make a synapse? So this shows the probability for the possibility of making a synapse. It does not show if it does really make a synapse. It just tells what is the problem. I mean, to make a synapse, they must get close enough to make one. So this is something I should say. Now, in your case, something which I would like to know is, when you say that two neurons are connected, you say it because you do it electron physiologically. Now, if two neurons make only one synapse with each other, can you be sure that you detect this? If one looks with electrodes for neurons which are connected, if they have several synapses with each other, you probably get a stronger signal as if they have only one synapse with each other. So the question is, could it be that the ones which are connected only via one synapse, that they are really detected always? Or
So usually the audience asks the questions.
Sorry.
So I think there are a lot of data out there in the literature, and my data aren't the best to answer that question. I think some of the best data to answer that question are from Bert Sockman's group, where they have experiments where they've looked at, for example, one particular paper looking at connections between layer 4 cells and layer 2-3 cells. And of course, like you say, if you don't detect a connection, obviously you can't say. But if they plot, for the cells where they actually find a functional connection that they can detect, and then they actually reconstruct the neurons and find the synaptic contacts, they find that the number of synaptic contacts in every single case that they plotted was either 4 or 5 synaptic contacts. So that suggests that the distribution is not probabilistic, that it starts off being, having the highest probability of 0 and the next highest 1 and the next highest 2. But the highest probability is 0, but then the next highest probability is 4 or 5. So it's not a probabilistic connectivity matrix in that respect, at least for those connections. And that's why I think that the connections that we and others detect when you stimulate and then look at the response, they're always highly reliable. And that's probably because an individual synapse is not necessarily very reliable. But since you have 4 or 5 of them contributing to it, it becomes reliable. But there are lots of data out there, and certainly ours are not the best to answer that question.
Yeah, I think that's something for a panel discussion.
Hi, Anand from IBM. What are the possible functional use for the 15% of the inhibitory neurons in the cortex? Any guesses or?
Somebody, also you, showed this morning how difficult it is to get an activity in that the cortex doesn't either die or explode. Because of the lot of excitatory feedback you have. And one knows from clinical studies that when that lack of inhibitory neurons leads to epilepsy, for example. So this is certainly one task of the inhibitory neurons to keep the equilibrium of activity. Then I think, again, this is an answer which Ed Callaway could answer very well, because I think he has a very interesting theory about functional differences of neurons, inhibitory neurons in layer 3 and 5, switching activity more between lower layers or upper layers. So also you can think of things like Inhibitory neurons are certainly also involved in shaping the receptive fields of neurons, for example, in shaping the orientation selectivity of neurons. So there's a number of functions one could mention. So they are certainly important. I just that I didn't mention them any further does not mean that they do not play an important role.
Okay, one last question, Fritz.
Almut, one question concerning the last part of your talk. Do you think, what about bidirectional connectivity between these patches? We know from the Van and Velleman work that that between areas, most of pairs of areas that are connected are bidirectionally connected. I always wonder, as a theorist and as someone who likes to simulate these networks, does this hold true also on the smaller scale of these patches that you see if you stain using the tracers? So would you expect one of these outcomes of your tracer on the other side also to project back to your injection site?
I would think so, yeah. I would think that much of this will be reciprocal. Sometimes most tracers are not completely anterograde or completely retrograde, and also in this the tracer we used, you often have a few retrogradely stained cells in some of these patches.
Is there data?
Sorry?
Is there some data?
Not from me, but indirect data, I think a lot. Not exact quantitative data, but.
I think we are out of time. Let's thank the speaker once more.